Imagine you have a very secret diary. You want a friend to analyze your writing style, but you don't want them to read the actual secrets. So you use a magic machine that reads your diary and spits out a long list of numbers, a numerical vibe that represents your writing, but isn't the text itself. You give your friend the numbers, thinking your secrets are safe.
But what if your friend is secretly a code breaker? And what if they can take that list of numbers and, with a bit of clever reverse engineering, reconstruct your diary entries almost word for word? Your secrets are suddenly not so secret anymore.
That exact scenario is what a new paper is exploring, but instead of diaries, it's about something far more personal, our DNA.
Welcome to Daily Papers, the podcast where we break down fascinating new research. Today we're looking at a paper called How Private Are DNA Embeddings? Inverting Foundation Model Representations of Genomic Sequences.
It sounds complicated, but the core idea is simple. We're in an incredible era of biology where powerful AI models, called foundation models, can read and understand the language of DNA. These models are trained on massive amounts of genetic data, so they can spot patterns related to health, disease, and ancestry.
And this is hugely valuable for medical research. A hospital might have genetic data from thousands of patients, and a research institute might have a powerful AI model that can analyze it. The problem, of course, is privacy. You can't just email someone your DNA sequence. It's the most sensitive, personal, and permanent piece of information about you.
Exactly. So a clever solution was proposed, called Embeddings as a Service. The idea is that the hospital, which holds the raw DNA, uses one of these foundation models to create a numerical summary of the DNA. That's what an embedding is. It's a long list of numbers that captures the important biological information without being the raw sequence of A's, C's, G's, and T's.
The hospital can then share this numerical embedding with researchers. The researchers get the data they need for their analysis, and the patient's privacy is supposedly protected because the raw DNA never leaves the hospital. It seems like a perfect win-win.
Well, the authors of this paper decided to play the role of the code breaker. They asked, what if we tried to reverse the process? Can we take those numerical embeddings and work backwards to reconstruct the original sensitive DNA sequence? This is called a model inversion attack.
And to test this, they took 3 popular DNA foundation models named DNA-BERT2, EVO2, and NT2, and used them to generate embeddings from human DNA sequences. Then they trained another AI model whose only job was to look at an embedding and try to guess the original DNA.
The researchers tested 2 different types of embeddings. The first is called a per-token embedding. You can think of this as a detailed, structured summary. The AI model first breaks the DNA sequence into small chunks, or tokens, and then creates a numerical summary for each chunk, keeping them all in order.
And the results for this first type were alarming. When using these per-token embeddings, the attacking AI could reconstruct the original DNA sequence with near-perfect accuracy. We're talking above 98%.
Let that sink in. Sharing this kind of detailed numerical summary is functionally the same as sharing the raw DNA sequence itself. The privacy protection it was supposed to offer was basically zero.
Okay, so that's a clear failure. But what about the second type of embedding? This one is called a mean-pooled embedding. This is a much less detailed summary. It takes all the numerical values from the different DNA chunks and just averages them all together into one single list of numbers.
Right, it's like taking every sentence in a chapter of a book, turning them into numbers, and then mushing all those numbers together. You lose all the order and a ton of the specific detail. This has to be more secure, right?
It is, but it's still not safe enough. The researchers found that while it was much harder to reconstruct the DNA from these averaged-out embeddings, they could still recover a lot of the original sequence, far more than you'd get by just randomly guessing.
And here's where it gets really interesting. They found that the length of the DNA sequence makes a huge difference. If you take a long piece of DNA and average its embedding, a lot of information gets lost, making it harder to reconstruct. But if the DNA sequence is short, the reconstruction can be scarily accurate.
How accurate? For short sequences of around 15 to 25 DNA letters, some of the models were so leaky that the attackers could reconstruct the original DNA with over 90% similarity. This is a huge deal because a lot of genetic analysis focuses on exact these kinds of short, functionally important regions of our genome.
So why are some of these AI models leakier than others? The paper suggests a key reason is something called tokenization. That's just a fancy word for how the model decides to chop up the DNA sequence into those small chunks, or tokens, before it does anything else.
They found that the DNA BERT2 model was the most secure, or the hardest to crack. Its trick was that it chopped up DNA into variable length pieces. It might make a token of ACG in one place, but TTAGC in another based on common patterns. This creates ambiguity, making it much harder for an attacker to piece the puzzle back together.
The other models were easier to crack. For example, the EVO2 model just used a single DNA letter as its token, A, C, G, or T. That provides no ambiguity at all. It's like trying to hide the words in a sentence by just separating the letters. It's trivial to put back together.
So what's the big takeaway here? This whole idea of embeddings as a service, which sounds like a brilliant way to enable research while protecting privacy, is actually full of holes.
Sharing detailed per-token embeddings is a non-starter. It offers virtually no privacy. As for the averaged-out, mean-pooled embeddings, they offer some protection, but they're still vulnerable, especially when dealing with the short DNA fragments that are often of most interest to researchers.
The paper concludes that we can't just assume these methods are private. We need to be rigorously testing them for these kinds of vulnerabilities. It suggests that tokenization strategy, how you chop up the DNA, could be an important factor in designing more privacy-preserving models in the future.
Ultimately, it's a crucial warning for the fields of AI and genomics. As we build these incredibly powerful tools to understand our own biology, we have to be just as dedicated to building the safeguards that protect our most fundamental information. Because once your genetic diary is out, you can't exactly get a new one.